please let me welcome my panelists, and thank you all very much for coming. You know, Ted, that experiment you mentioned, I wonder if there are young people here who want to hook themselves up to any. I'm Emily Yaffe. I'm a columnist for Slate. I write their Dear Prudence and Human Guinea Pig columns, and actually doing that experiment might be a good human guinea pig for me. I want to welcome my distinguished panel. I'm going to introduce them one by one, and they really represent a broad array of research being done in longevity, and they're each going to talk for a few minutes. Then we'll talk amongst ourselves and take questions from you. And as you can see, we have Anna Maria Cuervo, Aubrey de Grey, Stefan Johnston, and Cynthia Kenyon. I'm going to start with Cynthia. Cynthia is the American Cancer Society professor at the University of California, San Francisco, and the director of the Hillblom Center for Biology of Aging. She received a PhD from MIT, where she discovered that DNA-damaging agents activate a battery of DNA repair genes in E. coli. In 1993, Cynthia and her colleagues really upended our understanding of the mechanisms of aging when they discovered that a single gene mutation could double the lifespan of the laboratory worm they were studying up until that time. It was not thought that this could be controlled by a single gene, and she since then has been one of the leaders in longevity research. Cynthia, welcome.
Oh, thank you. So are you going to introduce everyone first? Should I talk now?
No, I'm going to
Okay.
give everyone a little introduction so we can remember.
Oh, so I am supposed to talk now. Hi, nice to be here. We had a great dinner last night. So as you just heard, I was also going to say we live a lot longer than we used to, but mainly that's because we're not dying from things that used to kill us, like infections. Okay, I don't know. Is it better now? Okay. Okay, is that better? All right, good. I'll move this over here. Okay. What? Is that still okay? Is it okay? All right. Okay. So there really has been no change in the fundamental rate of aging. And now that we're not dying of infections and so much of heart disease and other things, now we tend to die of Alzheimer's or other things that hit people when they're much older. And actually what I want to do right now, before I even get into the control of aging itself, I want to just digress and say just a few words about Alzheimer's disease, because I had some thoughts about it based on what happened at dinner last night, the conversation at dinner last night. Now, we spend $172 billion a year right now in the U.S. taking care of people that have Alzheimer's disease. And that's right now, and it's just going to get a lot worse as time goes on, as you know. In fact, when there was all this controversy about smoking, the tobacco companies were thinking of trying to make the argument that smoking might not be so bad for you, for the economy, because actually it was a lot cheaper to die of lung disease, of tumors in the lung, than it was to die of Alzheimer's disease. So anyway, people have been thinking a lot about what to do about this, and most of the talk last night was about how we can spend less on people that have Alzheimer's disease. But this is America, and I think we should think about research. It's amazing that even postponing Alzheimer's disease for just a few years could save a huge amount of money. And we spend, like I said, $172 billion taking care of these people. But how much do we spend on research per year? We spend less than one-half of $1 billion. 172, less than half of one. 469 to be exact, $1 million. So, and especially now, you can detect Alzheimer's disease in spinal fluid years before any dementia appears that you can see. So now we have a situation where you could actually test drugs. You could give people drugs that might be valuable for them early, before they actually are too late to cure or to treat. And like I said last night, Paul Greengard from a laboratory at Rockefeller, has very interesting compounds. There's interesting work going on at UCSF and other places. So I just think that in our conversation about what to do, we should really think about research. We really should, not just what to do about all these old people and how not to take care of them or take care of them more cheaply. Okay, so back to aging. All right, so for years it was thought nothing really could be done about aging. We just wear out like an old car, and it's a process of deterioration. And not only that, not only did it just look like something that just happened, there were evolutionary biologists who were making these arguments that you couldn't possibly have genes that controlled the rate of aging because they couldn't evolve because aging happens after reproduction. That was their argument. We could talk about that, but it doesn't really matter. For my point, the point is they had kind of put a lid on thinking about aging as a regulated process that could be controlled by the genes. But in the early 1990s, there were several labs, our lab included, myself included, who challenged this view. And so I just want to say just a few words about what we did. We looked for genes that controlled aging. So the idea is if there are genes that control aging, then if you change them, you could either speed up the aging process or slow it down. And if you just think about right now, there are turtles and fish that live to be 200, and they may not be able to play the piano, but they have perfectly good nerve cells and intestinal cells, all the neurotransmitters we have. So they're very sophisticated animals, and they live way longer than we do. So anyway, what we did is we took a little animal called C. elegans, which is a worm, but it is not an earthworm. It's a roundworm, and it's only the size of a comma in a sentence. I guess about a font 12. Anyway, so in those animals, it's really easy to change genes, and so we did this at random to ask, could we find a mutant? A mutant is an animal in which you've changed a gene, and the change in the gene is called a mutation. So could we find an animal, a mutant animal with a gene change that lived longer? So we did this, and we found that if we change one gene called DAF2, DAF-2, that we doubled the lifespan of the animal. So all the other genes were exactly the worm has 20,000 genes in it, and all the rest were fine. They were the normal genes, and you change this one, and now it could live twice as long, and all the other genes must have been able to keep it going for twice as long as they normally would. And the thing that's so amazing about these animals, and it's really hard to get your head around, is that they stayed young longer. So, for example, it's kind of like, you know how your dog ages quickly? You can just watch your dog age. And then you can get another dog and watch it age. So it's kind of like turning a dog's rate of aging into something that's more like that of a person. It's not a 7-fold increase, although we've done that in the lab now. We have animals that live 7 times longer, which is more like a dog to human, but it's kind of like that. So here's another way of thinking about it. Suppose you are in your 40s, you're single, and you're dating, and you meet someone, you like them a lot, very attractive person, you get to know them, and you take them out to dinner, and then you ask them how old they are, and they say, well, actually, I'm 80. That's what it's like. It's because their age, it takes them 2 days to age as much as a normal worm ages in 1 day. And I keep beating on this because people always think, even after I finish talking to them, if you start talking to them, like I give a talk like this, and if I start talking to someone in the break, you can tell that what they're thinking is that you have a way of making a 90-year-old person pretty healthy, but still looking like a 90-year-old person. And it would be like a 90-year-old person looking like me, or someone younger than me. So anyway, that's what we did in the lab with these little worms. So what did we exactly do molecularly? It turns out that when we changed this gene, we perturbed a system that the animal has that senses nutrients and stress. And specifically, we changed the tissues so they could no longer respond to hormones that are similar to the human hormones called insulin, which you know about, and IGF-1, which you may or may not know about. Now, insulin is known to be required for the tissues that take up nutrients after you eat a meal, and IGF-1 promotes growth. Now, these proteins, these hormones are active when everything is fine, when you're eating a meal, when you're growing, when you're pregnant. For your baby to grow, the IGF-1 is promoting growth. The loss of these hormones kills you. It kills the worms, it kills everybody, it kills people, it kills any animal, that you absolutely have to have these hormones. But what's interesting is that just having a little less of the signal, what happens is very kind of unexpected. It's neat and it makes sense in retrospect, but here's what happens. If you have a little less, the animal senses that there's danger. It has a danger signal, a signal that might be similar to getting less food or feeling some kind of stress in the environment. And what it does is it responds to that stress, and it rolls out a protective response. And it does so because there's a protein called DAF16 in worms, and the same protein is called FOXO in people, same protein but different names. The protein that goes into the nucleus and activates all these protective genes that can extend lifespan. They do all sorts of things. The genes encode proteins that help bolster the DNA repair capacity of the cell. They encode antioxidant proteins. They make the immune response better. They encode proteins called chaperones, which help other proteins fold correctly. And if the proteins are damaged, the chaperones escort them to the garbage can so they can be recycled in the cell. Cells have garbage cans, actually. It's pretty cool. Anyway, so you can think of FOXO as like a building superintendent. Okay? FOXO is the gene regulator protein. So basically, when things are good, he's a little lazy. Building is not so bad, but it's deteriorating a little bit. And then suddenly, he realizes there's going to be a hurricane, or the owner of the building is coming. So he thinks, oh, there's danger. And so he hires a team to come. He doesn't do it himself. He hires a team to come and fix things. And they all do different things. There's a window repair person, a floor person, a roof person. But they all do their own thing, and they all do it at the same time, just like FOXO does, to keep the cells in good shape. And by combating damage, you combat one of the major causes of aging, and you extend lifespan. So, okay, so that's what's going on in these little worms. Now, what about other organisms? It's just a worm thing. By the way, this work is all funded by NIH. Not to say that the Defense Department wouldn't fund it, and I think the Defense Department is great to fund research, but NIH is doing a great job. That's what I was saying last night. Anyway, to those of you who were there. Anyway, so let's see. Right, other organisms. So it turns out the way science works these days is people already know that if you make a big discovery in yeast or worms or some very humble little animal, chances are the same thing is going to go on in a higher organism. That's been proven again and again and again for many different processes in biology. So it's great because you can use these little simple animals and really fast find interesting things, and then other people can test them in animals like mice that are more difficult to work with. So sure enough, if you change the same genes, if you just damage them a little bit in flies or in mice, you extend lifespan. And it looks like the same genes also affect human lifespan as well. So for example, there are several different reports of people having mutations or altered forms of the genes in the IGF-1 or insulin pathway, specifically the IGF-1 pathway in humans, that have exceptional longevity. In other words, people who tend to live to be 100 are more likely to have gene changes in components of this pathway I've been talking about. And really the hero of this is the FOXO gene. There are now 7 populations all across the world in which variants in the FOXO gene have been shown to be associated with the ability to live to be 90 or 100. And so these include the Chinese, Germans, Italians, Californians, New Englanders, Hawaiians of Japanese descent, everybody. Well, I don't know if everybody, but those people at least, which is a lot of people. And so I think what that tells us is great. I think it tells us that humans are more than likely susceptible to the effects of this signaling pathway on lifespan. It means there's something we can push on. Doesn't mean it'll really work, but it's something, it's there. If we push a little bit, we should get a response. So the other thing that I think is really interesting, as you heard in the introduction, everybody's thinking, well, if we live longer, we would just get Parkinson's disease, we would just get Alzheimer's disease. Well, actually, the amazing thing about these gene changes, which keep animals young longer, is that they make them resistant to a whole variety of age-related diseases. So, and it kind of makes sense, if, in a way, if an animal is, like if you're on that date and someone looks like they're 40, you wouldn't expect them to get Alzheimer's disease because they look, and they don't, at least in this, if they were worms, at least I'm making an analogy to the human, because we don't have that yet in humans. But we do know even in mice, changes in these genes, which extend lifespan, also delay Alzheimer's disease in mouse models for Alzheimer's disease. So this is a way we have now not only potentially of staying young longer and maybe productive longer, hopefully, but also of delaying a whole variety of diseases all at once. I mean, aging is a way bigger risk factor for cancer than smoking. You're 100 times more likely to die of a tumor if you're 60 than if you're 30. So it's a huge risk factor. So if we go after this, there's all sorts of possibilities. So what are people doing about this, and what are the prospects? Well, our lab now is trying to find small molecules that will activate FOXO in culture. I'm just about done.
Yeah,
2 more sentences.
and what you don't get to, we can...
Okay, just 2 more sentences. Mm-hmm. Okay. So that's what we're doing. We're going after FOXO. There's a drug called rapamycin that targets another nutrient sensor called TOR. And if you give mice rapamycin, even when they're old, it slows down their aging and extends their lifespan. This is an FDA-approved drug. I'm not sure you should take it because it can affect the immune system in ways that might not be good for you, but it's definitely something to think about. And there's other drugs that target other genes that we can talk about later that are now in trials. Again, for age-related diseases, but they may have effects on aging per se. Okay, so it's a bright potential. And especially if NIH doesn't go bankrupt.
Thank you very much. Our next lab scientist is Ana Maria Cuervo. Ana Maria is the professor of developmental and molecular biology and co-director of the Institute for Aging Studies at Albert Einstein College of Medicine. The focus of her research is the role of protein degradation in neurological diseases such as Alzheimer's and Parkinson's, etc. She works on what are the housekeeping mechanisms of the cell. In the recent book by Jonathan Weiner, Long for This World, in which he discusses the work of Ana Maria, he says housekeeping doesn't get much respect in life and doesn't get the respect it deserves in the laboratory, but she is considered one of the leaders in the field of protein degradation, which many people think may be the key to understanding the mechanisms of aging. So, thank you.
Thank you very much. First, I want to thank the opportunity to be here. And as you mentioned, we work in protein degradation, or if you want, you can call it the garbage containers of the cell, as Cynthia reflected to, because we are all about garbage and cleaning. And the first thing that I want to get across is that we are mostly, we are talking about different aspects of aging, and you have heard about genes from Cynthia, and you will hear about cleaning from me, but these are not contradictory points and reflect that we are going separate ways. One of the important things of aging, and I think all of you are aware, but just to emphasize, is that it is a multifactorial process. There are going to be many factors that are going to influence the way that you age, and there is a lot of room for intervention. And also the beauty is that all these different factors are really interconnected. Small modifications in a gene are going to affect the cleaning systems and many other pathways in the cell, or modifying these cleaning containers is going to preserve better your DNA, so your genes are going to be better. So don't think about like the different areas of research in biology of aging as different people pulling in different directions, but we are really unified in the whole idea that this is a multifactorial process, and also with the idea that, and I think there are good examples now in the literature, that modifying one of these factors, it has a big impact in all the others. So by doing very little, you can get a lot out of the cells. And then the other point, before I tell you a little about what we do in the lab, that I want to emphasize again, and that we discussed last night, is that I know that there is going to be later a discussion about, okay, what are we going to do with all these old people, and they are very expensive for the society, they have Alzheimer's, but the whole idea is not to keep people longer with those diseases. It's going to be cheaper because in a way they are not going to have those diseases, and then they can go on those dates and enjoy dinner, as Cynthia was mentioning. So it's going to be, the idea is to achieve complete functionality and independence, and this is and be free of disease, and this is supposed to have a very positive impact in the society. But the way that we are approaching this, and as I said, there are very different ways, we are mostly interested in damage and repair. And as you know, all the cells in your body are continuously exposed to damage. And I'm not talking only about the environmental and the contamination outside and all those things, but also from your own body. And this is where the connection goes with the food and the nutrients that I'm very sure it's going to come out through the discussion. So for your cells to produce the energy that they need to keep functioning, they are going to have to generate byproducts. It's like any factory generating something, you have byproducts, and those byproducts you hear then as free radicals and many other names, basically are going to damage structures in your cell. But this is not a problem. This happens since you are born. This is happening today and continuously, and your cells are well prepared to deal with that damage. You have quality control mechanisms that are going to be able to identify what is wrong. For example, chaperones that Cindy also mentioned, and are going to identify, okay, this is a bad protein, or this is a bad organelle, and I just have to eliminate them. Or I have to repair it. And sometimes the repair is possible, and sometimes it's a little like an old car. If you repair it today, tomorrow is going to be a different thing, and then next day the other thing. And the cells do pretty much what you do with your car. You just send it for pieces that then can be reutilized, and that's the beauty. There is a recycling process there, and then you just get a new one. And this process happens continuously every day in every cell in your body. So one of the things is that if this cannot happen, or as you get old, the activity of this cleaning mechanism or recycling mechanism really decrease. And the approach that we are taking is that the accumulation of that damage is in part contributing to the malfunctioning. So what we've been trying to do in the lab is trying to understand a little more about these garbage containers or about these recycle systems and trying to improve their function. And by improving, I don't mean to create like super cleaning cells. I mean, basically you just have to go back to the levels that they have when these animals or these systems were born. So we work in the lab with mice, and basically the whole idea is trying to identify why these systems don't work and what can we do to fix it. And a couple of years ago, we identified for one of these cleaning mechanisms that there was a defect and there was this decrease in the levels of a particular component of the cleaning machinery, and we just restore it. And we use genetic manipulations in mice to put this particular protein at the same level that when the animals were born. And the interesting thing is that then we found that if you do that in an organ and in an old animal, the organ is cleaner, but it also functions better. And this is what is all about aging. I mean, you don't want just to maintain them for longer. You want it to return to functionality, and that's what we found. And I have to say that this was really surprising for us, that just modifying one thing, you have such a big impact. And I think I can say, I mean, this is just a cleaning system, but I can say, as my mother always say, in a clean house, everything works better. So it kind of also applies to your cells. All of a sudden, your genes are going to be less exposed to damage. Your other systems in the cells are generating and being more efficient, generating energy. So you can have a big impact with a small modifications. And the same that I'm saying for cleaning mechanisms, you have it for other process. You can just affect one of them, and you have kind of a rheostat that put you back in a level of homeostasis. You are in a better conditions, your cells are going to be more functional, and this has to improve general function of the organism. And then the idea, of course, for us and the future is not to do gene therapy in all people. I mean, this was a proof of principle that you modify this, you can do these manipulations and restore normal function. But the idea is what Cynthia was also pointing to, is now trying to identify molecules, compounds, interventions that can do the same without having to go for the gene therapy approach. And actually, there is a lot of development. And then the interesting thing, and going back with this idea that many factors are interconnected, is that the same drug that Cynthia was referring to, for example, rapamycin, that has this effect in all of the other functions of nutrient sensing, it's also known to activate this cleaning mechanism. So you can identify through this kind of studies and through this very, very basic research, you can identify the interconnections in pathways like cleaning and eating that initially you might not see the connection right away, and also find molecules that have the capability to activate both mechanisms or even more. And the whole idea, as I say, is not for you to go out and try to start drinking rapamycin. I mean, you need the research to continue to come out with more specific molecules that can really separate these functions and can target them in a different way. But I have to say that the future on that respect, I think, is very promising because, as Cynthia mentioned, there are already drugs there is already interventions in mice, and then also the other important thing is that most of these interventions that are prolonging lifespan, even in mice, they are not just acting in making the animals to live longer, but the same compounds have been shown to delay the onset of Alzheimer's, cancer, and many other infectious diseases, and many other pathologies that you have associated with aging. So basically the whole idea is that you can act in a system but have a major impact in the whole function, and I think this is possible and is happening. And now the major limitations are, in one hand, to continuous identifying the interconnections between these processes, and that's what we do in the labs, the basic scientists. You also need a better way to develop these molecules, and of course that costs money. And I'm not asking for money here, but at the bottom of line, yeah, we need that. But also, in addition, I think we have to start thinking in the translation to humans. Most of our studies are in worms or are in mouse, and you are starting, and now there is a momentum to start doing interventions or studies in humans that are not invasive, that you can start collecting this information and then start testing if this is also functioning. And I think this translational component is going to be very important in the next coming years, because this is going to give us the idea that whether or not you can affect all the systems. But I have to say that the future looks very promising.
Thank you very much. And that was a perfect segue to Stefan Johnston, who's all about translational research. Stefan is the director of the Center for Innovations in Medicine at Arizona State University's Biodesign Institute. He's the author of over 150 journal articles and has 20 patents. His work focuses on innovative solutions to fundamental problems in biomedicine and bringing them to us cheaply, safely. He's going to talk a little bit about some of this, but for example, he and colleagues are working on a universal vaccine to prevent cancer, and he's also working on a system in which people would be very cheaply able to monitor their health on a daily basis and identify pre-symptomatic disease to be able to prevent having disease. Why don't you think big, Stefan? Stefan Johnston is going to talk about how he's going to make us all healthier.
I don't know if I'll do that, but first of all, when I first heard about this meeting, I was thinking that I'm not sure I agree with the basic tenet. It was called radical life extension. And I've known a lot of radicals that I'm not so sure I'd want to extend their life. And so, but then Joel clarified what this really meant. And so I was asked to talk more on the product side of what might be coming down the pike. And I basically see I see this in three phases, but I have to say, you have to take everything I say with a grain of salt, because last night I tried to prove to everybody that you can't predict the future, and you shouldn't believe anybody that's trying to predict the future any far out. But so with that said, here's my 2 cents. I think the first phase that will have a big impact on life extension and quality will be where we learn to detect disease, chronic diseases early, and then treat them much more effectively. So the basic idea is that we begin to have the point where we can monitor the health status comprehensively of everybody, inexpensively and simply, and then detect disease early, especially chronic diseases like cancer and Alzheimer's and others, but including infectious diseases, and then treat them, and then that early detection will drive a new paradigm in the types of treatments that we have. They must be much more targeted and much less toxic. And I think that that's certainly conceivable, and that's conceivable on the short range. And last night I tried to demonstrate a technology that was a prototype for that type of approach we call immunosignaturing, where you can just take a drop of blood, put it on a filter or saliva, send it through the mail, and then we can get a signature of your antibodies, and those antibodies are basically quite revealing in terms of your health status. And in fact, I think we might have a few of those prototype kits in the back that people can use if they want to see what it is. And then the next phase beyond that will be where we actually not just detect disease early and treat it, but actually prevent it. And we've heard the prototypic types of ideas about how to do that here, where things probably much more sophisticated than rapamycin will be designed that people take and actually prevent disease from happening in the first place. And that's obviously much more desirable. On our part of that, we're not working on the drugs that can manipulate the aging system, but as Emily mentioned, trying to develop a system to prevent cancer from happening in the first place, trying to treat cancer like it's an infectious disease. Probably the most effective medical interventions that we've ever done as human beings have been vaccines. The rate of return on dollars spent for life saved is immense. And so we think that you can actually treat cancer like it's a prophylactic disease, cancer infection. And then finally, I think we'll probably go to the next phase, and this part we aren't doing anything in. And that's where we not only have extended our biological selves, but begin to merge more and more with artificial intelligence and robotics. That eventually we transfer from being people, just simply biological people, but robotic people. And this will happen. It's already happening. People have cochlear implants. People have parts of their heart replaced. This is happening in subtle ways now, but it may happen much more dramatically in the future. Computers already have the computing capacity of our own brains now, and they compute at 1 million times faster. So the future for computers to meld with us is probably not that far off. But as I said, take everything I say with a grain of salt. Thank you.
Thank you. And our final panelist is Aubrey de Grey. Aubrey is the chairman and chief scientist for the Methuselah Foundation, a nonprofit charity dedicated to combating the aging process based in Cambridge, England. He's the editor-in-chief of the journal Rejuvenation Research. He's the author of the books Ending Aging and the Mitochondrial-Free Radical Theory of Aging. He's a provocateur, pushing laboratory scientists to come up with the fixes so that he will be able to live to 1,000 and attend many, many more seminars. So, Aubrey?
Thank you, Emily. Thank you to the organisers for having me. I'd first of all better correct the introduction because I'm no longer the Chairman and Chief Science Officer of the Methuselah Foundation. I'm actually the Chief Science Officer of a new foundation called Sense Foundation, which is what has been built around the ideas that I'm going to be describing today. But don't worry about that.
Explain what SENS stands for, because I think that's interesting.
No, it's too long. I also want to thank Stephen for only taking 5 minutes. I might take a few of those minutes myself. I just wanted to start, very much as Stephen did actually, with a comment on the title of this meeting. I think that it perhaps needs to be mentioned at the outset that the whole concept of radical life extension is a bit of a turn-off for a lot of people, including, I think, a lot of people on Capitol Hill. And so I want to make sure that everybody understands what this is really about, that we are actually talking about hard science, hard technology of the relatively foreseeable future that will keep people healthier for longer, and that any side effect in terms of longevity should be viewed as exactly that, as a side effect. The concept of the myth of Tithonus that was mentioned in the first talk this morning is something that we need to get out of our heads. People will not be living in a frail, diseased state at the end of their lives for a lot longer than they do today, whatever interventions we develop. There may be small variations, shrinkage or expansion of that period of life at the end of life, but it will be small differences. We will not be keeping people in a state of ill health for a radically extended period. And the first thing I want to say, actually, in comments on the first talk we heard is, whatever you do on Capitol Hill, please don't listen to Guy Brown. Guy Brown wrote this really very influential book called The Living End, which has started from the precept that people of a given chronological age are more or less as healthy and no healthier today relative to, let's say, 50 years ago. So 60-year-olds today or 70-year-olds today are pretty much in the same state of health as 60 or 70-year-olds of 50 or 100 years ago. I don't know any actual gerontologist who would agree with that statement. So please do not take into account anything that might be concluded from that assumption. It's an incorrect assumption. We are much healthier now at age 60 or 70 than we were. So I want to start, spend a couple of minutes commenting on the remarks that were made by Cynthia and Anna Maria, who are doing, as you've heard, some of the most important cutting-edge work in understanding how to control aging. So Cynthia's work and the work of other labs on the identification of simple modifications, whether genetic, such as the DAF2 gene that Cynthia mentioned, or pharmacological, such as rapamycin, these interventions are highly remarkable, and certainly there was a great deal of surprise in the field when it became apparent that such interventions existed, because aging, let's face it, is an extremely complicated phenomenon. An awful lot of different things go wrong in the body over time, and it just doesn't seem very likely that you would have a simple intervention like that. But we now understand fairly well why there are such interventions. It's because the process of evolution has created a whole bunch of really complicated machinery within the cell and within the body that allows the body to respond to environmental changes, and we can mimic and exploit that machinery by making simple changes that are not exactly the same things that would have happened environmentally. The main environmental change that I'm talking about here is famine, deprivation of nutrients. The worm that Cynthia studies is capable, if you catch it at the right time during its development, of entering a very strange state called the dour state, which allows it to live several times longer than normal. And there is a lot of reason, a lot of science now that shows us that the genes that are most effective when you mutate them in extending the lifespan of this organism are actually genes that are heavily involved in that pathway. So essentially what's being done here is the turning on of a switch, effectively, that activates a whole bunch of machinery that's already present in that organism. Now, of course, the big discovery that Cynthia and others made was that you can turn on the life extension in these worms without turning on the other changes that turn the worm into some kind of spore. And so this is a fantastic discovery. But the point is, it is the activation of machinery that's already present in the organism. And that's quite a downer when we think about how it might relate to humans. Because, first of all, let's look at mice. We can't make mice live 7 times longer than they normally do, irrespective of what genetic or pharmacological things we do. Now, one day we might be able to discover how to do that, but we can't do it yet. And we've been trying for quite a long time, and we haven't got above 50% increase in the lifespan of mice, even from interventions that are done transgenically or in the in the parents of the mice in question. So intuitively, a non-scientist might get rather pessimistic and say, well, okay, maybe it's just that longer-lived species just don't have such good life-extending machinery built into them that can be switched on simply. And actually, there turn out to be good evolutionary reasons for expecting that that would be so. So I think the onus at the moment is on scientists to try to really max out what we can do with relatively long-lived species compared to worms, like mice, for example, species that are more closely related to us. And the more that we continue and fail to do that, the more that we are confirming the intuitive pessimism that I think a lot of people on Capitol Hill and elsewhere have with regard to the ability to actually postpone aging by simple means. We heard about rapamycin just now, this drug that succeeded in extending the lifespan of mice. Now, you shouldn't get too excited about that, because the lifespan extension is only about the same as one can achieve by the very simple dietary manipulation, nutrient deprivation essentially, without malnutrition. However, I do want to say one thing about the most famous aspect of the rapamycin life extension result, which is, as Cynthia briefly mentioned, the fact that it works in mice that only receive the drug starting in middle age. This is a spectacularly important thing, which obviously makes a great deal of difference from a policy point of view. If we could actually do stuff to people who are already in middle age and significantly postpone their ill health, their age-related ill health, then that would be rather good news for the baby boom generation, which is, of course, the generation that policymakers and economists and so on are most interested in helping right now. And so the big thing I want to mention about that experiment was that it was done by accident. The original intention was to feed this drug starting at a much younger age, and for complicated reasons that I won't go into, it only was started in middle age. And the reason I want to mention that is not because scientists are not interested in helping people who are already in middle age. It's because the whole structure of funding of work in intervening in aging has very severely neglected late onset interventions. If there's one thing I want to recommend with regard to change that could be made today in the prioritization of which experiments are done, it is to put much more emphasis on the exploration of interventions that start late in life in laboratory organisms, because the promise of those interventions is much greater than is popularly perceived. However, what this says is, we're not actually seeing a way to demonstrate to the public that we can greatly postpone the ill health of old age, especially not if we start late. Before Cynthia and other labs discovered these simple interventions that could extend life, there was actually some other work that was done that got the same sort of degree of life extension in a laboratory organism, in this case the fruit fly. And it was done in a manner that did not overturn dogma within the field at all. It was actually a confirmation of what was already thought, because how it was done was by selection. Over multiple generations, the creation of long-lived fruit flies starting from relatively short-lived fruit flies, and the degree of life extension was about the same amount, roughly a doubling in the first instance of longevity. But of course, that's not so much use for those of us who have the misfortune to be already alive. So, again, we're not really seeing a way to convince policymakers that this work is as valuable as we think it is. So now I come to the question of, what's the way out of this? Can we, in fact, explore the option of radical postponement of age-related ill health? And I think we can, but the way that we're going to do it, in my view, is by exploiting the science that has been developed in other areas of biology, not predominantly within gerontology, namely the area of regenerative medicine. So cars have been mentioned a good deal already as a good analogy. The human body is a machine. It's a really, really complicated machine, but it's still a machine. So maybe it makes sense to think about, postponing ill health in people by looking at how we already succeed in postponing ill health in simple man-made machines like cars. And of course, the way we do it is actually not by changing the way that the car functions. What we do is periodic repair and maintenance. We take the car into the mechanic every year or whatever. And we know that if you do that only as well as the law requires, it doesn't work very well, it doesn't work for very long. But if you do it as well as people who love their cars do, if you do it more comprehensively, you can keep cars going indefinitely. That's why we have so many 50-year-old VW Bugs driving around the streets of this country. It's why we have 100-year-old cars that are doing just as well as when they were built. So it's possible that we might be able to apply regenerative medicine to the problem of aging. Now, I want to emphasize that this is far, far more difficult than the approaches that you've heard about so far, these simple genetic or pharmacological manipulations. But just because it's so difficult doesn't mean we shouldn't think about it. Getting to the moon wasn't very easy either, but we did it. And if we can pull off a regenerative medicine approach to combating aging, a sort of divide and conquer approach to repairing the various types of molecular and cellular damage that accumulate throughout life and eventually kill us, then the potential impact in terms of postponing age-related ill health is vastly greater than anything that we have any reason to believe we can achieve by simple interventions. We should, in particular, be able to radically postpone age-related ill health in people who are already in middle age before we start. And that's the really important thing to remember about all of this. So in the last minute or two of my remarks, I wanted to focus on a couple of things of particular relevance in terms of policy and in terms of economics of all of this. The first thing is that, again, I want to come back to the title of the conference and perhaps the slightly incorrect title of the conference, or at least misemphasized. We do not want to be focusing on radical life extension when we think about all this. It's the health of people that we're talking about. Radical life extension means the extension of something that's going to happen in the distant future. We are not going to have anyone aged 1,000 for 900 years, whatever happens. People only get older at 1 year per year. The change in the age structure of the population is relatively gradual compared to the changes in society and economics and so on that happen as a result of other technological change. So we really don't want to be focusing on that. What we want to be focusing on is the distribution of health within society that will occur if we succeed, even modestly, in postponing aging. And I do want parenthetically to point out here that I very strongly support the work that's being done by people like Cynthia and Anna Maria, which even though it may only very modestly postpone aging, that's a great deal better than nothing. It could make an enormous difference to the economy. And so I think it's absolutely essential to work on these simple interventions as well as on the much more ambitious, longer-term interventions that might be able to do a great deal more. The health demographics of the population is what matters most. The final thing I want to mention is in terms of time frames. Now, I mentioned that the regenerative medicine approach to combating aging is really difficult. I think we only have a 50% chance of really decisively developing such interventions within 25 years. We sure as hell aren't going to develop them within the next 10 years. So you might ask, if you're a congressman, for example, you might ask, why should you care about this? I'll tell you why. The reason you should care is because people are going to start caring about this, not just when these new medicines arrive, but when they become widely anticipated. In other words, when results in the laboratory applying regenerative medicine comprehensively to laboratory organisms, especially to rodents, actually deliver substantial life extension, perhaps 5 times as much as we achieve with rapamycin started in middle age. Once they start doing that, scientists are all going to see that regenerative medicine is a practical solution to the problems of aging, and they are going to say so. And more or less overnight, it's going to cause a very great deal of change in the way that people think about their future, in people's priorities, people's spending priorities, and people's voting priorities. And that may very well happen within less than a decade, perhaps much less than a decade. So that's why you really need to think about this. Thank you.
Thank you very much, panelists. Let me start by asking, several of you talked about interventions in animals in middle age. There are probably some people in this crowd here who wonder, are there any interventions that people, humans, not mice or nematodes in middle age, can do now? Is your research applicable to us in any way? We've all probably heard about calorie restriction, and from the looks of you, maybe you're all doing that. There's Resveratrol, which is a component of red wine. I understand we can't drink enough to make us live forever, but it's available as a supplement. Are there things we should be doing now that come out of your research to at least extend our healthy lives?
Well, I have an answer.
Please.
Okay. First of all, let's get one thing straight about resveratrol. Mm-hmm. Resveratrol extends the lifespan of mice that are fed a high-fat diet, that are sick mice and would otherwise die early. But it doesn't.
That sounds like most Americans.
It does not. Well, that's true, but not all. But it doesn't extend the lifespan of mice that are just ordinary mice fed a normal diet that aren't fat. It seems to make them healthier, but it doesn't extend their lifespan at all. So, okay, not that it might extend human lifespan, but we don't have a precedent for that from mammals. So I don't really know the answer to this question, but I told you about the fact that the insulin pathway regulates aging in insulin and IGF-1 pathway in worms and flies and mice. And we found this FOXO gene that I told you about, which the FOXO protein is the gene regulator. It's the building superintendent I told you about, which springs into action when it thinks there's danger. So that protein is present in normal worms, and it actually makes them live longer than they otherwise would. If you take it away in an otherwise wild-type worm, now they age faster. So what would regulate FOXO? What would control its activity? Well, insulin would, because insulin regulates, as I told you, FOXO, we already know that. But what regulates insulin? Well, glucose regulates insulin, sugar. So when you eat sugar, your insulin levels rise, and that depresses FOXO activity. And we found that if we gave our worms just a little bit of sugar, they eat bacteria normally, which are tiny little single-celled animals. But when we give them just a little bit of sugar, 2%, which is, and now a candy bar is 75% sugar, so this is not very much. 2%, they age faster and they live shorter. But if we took a worm that we'd already deleted the FOXO gene, so it already had a short lifespan, it already aged fast, and now we gave that guy 2% sugar, it didn't live any shorter. So that suggested very strongly, and other evidence that we found in the lab, that sugar shortens lifespan actually by doing the reciprocal of the things that we did to extend lifespan. So there's a lot of data now suggesting that a low glycemic index diet can be good for you. A low glycemic index means eating food that isn't turned into sugar very rapidly in your body. If you stop eating right now, and then you starve to death over the course of the next couple months, after about 4 hours after your last meal, your body keeps the level of sugar at about 100 milligrams per deciliter, all the way till death. It breaks down glycogen, then it makes its own sugar and so forth, but it stays very constant. But after you eat a meal, especially a Western meal, the level of sugar in your blood goes up 4 times or more. It's huge. So if you eat a low glycemic index diet, you're eating a diet where the food that you eat, the sugar in the food is released slowly. Either there's not sugar in the food you eat, like if you eat fat, or if you eat broccoli or something, there is sugar there, but it takes a long time for it to get out. So I think that kind of meal, I know it's not bad for you, especially if you eat enough green vegetables and you don't just only eat fat. You eat a balanced meal, but you cut out the sweets and the starches for the most part. I think that there might actually be some benefit, certainly for health and possibly for longevity. It's not proven, but I think there's some unpublished data from monkeys that you'll learn about before too long, but I can't mention it here. It's not from my lab.
Anyone else, things we can do now or things that appear on the horizon that might actually help the people in the room?
Right. Well, I think I want to come back to Cynthia's first point, and your point, which was, yes, what we can very probably do is increase the health, especially of most Americans today, because the overeating that has become so prevalent in society, and of course it's not just America, but I think America probably has it worst, is an enormous impediment to good health in old age. And the more that we can do to stop that happening, the better. It may very well be that the most effective way for most people to actually cause that to happen will be pharmacological, or in other ways other than simply eating less. For the moment, however, I recommend eating less. And of course, there are other things that you already know. Don't smoke. But in terms of actually improving the health and the healthy longevity of people who are already looking after themselves and behaving the way their mothers told them to, I don't think that at the moment we can do very much. Now, there is, as I mentioned earlier, a great deal of merit in doing our best to develop ways to do this. But as Cynthia mentioned, Resveratrol does not seem to have to be the magic bullet that can substantially postpone age-related ill health or indeed death in mice, and therefore probably not in humans that are being fed normally. Of course, there is a great deal of work going on, especially by Certris, the company founded by the people who discovered the resveratrol phenomenon, to develop analogues that might work successfully in normally fed animals and normally fed humans. But it remains to be seen whether that's actually going to happen.
So I don't have any solution, but I have sort of an evolutionary perspective on this, which is interesting, is that last year was the first year that there were more overfed people than underfed people in human history. And it's quite remarkable, but it makes sense. I mean, we as humans, we evolved genetics, and most of our evolutionary history was driven by genetic evolution. And it was only over the last few thousand years that our evolution is driven by our technology, not by our genes. But we still carry that baggage. So for most of the time when we were humans, it made sense to grab every calorie we could. And we developed tastes that made sense to grab every calorie. So if there was sugar, get it, that's immediate energy. If there was fat, get it because it's high content energy. And that's what we're still burdened with, that genetic history. We're just going through an evolutionary period where we haven't adapted. For the first time, we can provide that amount of food for ourselves. Remember, for most of our history, we were either hunter-gatherers or, up until relatively recently, most of our population was engaged in making food. Now 2% of our population feeds our country. And so this is the first time we've had this, in evolutionary history, we've had this kind of overabundance capability. And so I just look at this is an adaptive period. This is a relatively brief period in our evolutionary history that we're going through, and we'll adapt to it because we're so adaptable. And it'll be behavioral adaptations, and it'll be technological adaptations. And they'll probably have quite dramatic impact on our health and probably our longevity.
So I guess my 5 cents comment is also in the direction of the nutrition. I agree that we are not now in conditions of using some of these drugs to expand, but by the same criteria, one of the best activators of the cleaning systems in your cells, and going back to the garbage containers, is actually reduce the caloric intake, and glucose has been shown to have a very negative impact on high-fat diet and all these factors. We have papers published how much they decrease the activity of these cleaning systems. So once again, it's a single intervention that is going to add at very different levels.
So most Americans are not going to look like this panel. You've talked about your laboratory work and how promising that is. Do you have a time frame for translating this to actual interventions? Ted Fishman talked about he was going to a Parkinson's meeting, and I know if you're interested in Parkinson's disease, probably people have been told for 2 decades who are diagnosed with it, hang in there. This is the model disease for something we're going to fix. And I was talking to a Parkinson's researcher for a story, and he said a lot of my patients are getting quite bitter because they say it's been 15 years, 20 years since I've had this, and there's really nothing. So is this all, do you see a 20-year timeline for translating this research into interventions? 5, who knows?
So I guess since I'm the one doing a little of Parkinson research, so I think it's difficult to put a number, and I think everybody will agree on that, and also depends on the money that we get to do things. But the most important thing is, for example, in cases like Parkinson, I think the important thing is that there has been a change of mentality, and that's why they are inviting people like Ted to talk about this, because so far there has been this tremendous emphasis in looking for the genetic cause and try to understand the system. But then, similar to cancer and other diseases, it has become clear that the biggest risk factor is aging. So now the emphasis have to go into the contribution that aging is doing to that disease, because, for example, even the genetic forms of Parkinson, somebody who has it because it inherited, is born with the disease, is born with the abnormal protein that accumulates in their brain, but his body is able to deal with that. So if you can maintain that level of quality control over that protein, or the right combination of pathways to maintain it properly, or even use artificial methods to maintain homeostasis as it was when they were born, that should be kind of the intervention, rather than going for the single individual factors in each of them, because once again, aging is the maximal aggravating factor here.
All right. Yes. Okay. So I wanted to say, to address your question, but it's to start off by also addressing something that Aubrey was saying. He was implying that the best we could ever do by changing what the body, encouraging the body to express its own life-extending potential would be very modest. That's possibly true. It's also possibly true that regenerative medicine could make us immortal. That both of those are possibly true. However, I just want to point out that in evolution, going from a lifespan that the worm would have, our little C. elegans, which is probably more or less what some of our ancestors, distant ancestors had, which is a couple weeks, to a lifespan of 100 years, is a 2,000-fold lifespan extension. And that happened by mutations, by changing genes. Just like the DAF2 mutation is a mutation that changes the gene and doubles the lifespan, there must have been other gene changes that happened, and maybe even those that happened during evolution. And there are long-lived and short-lived birds, mammals, and insects. So that means evolution didn't just extend lifespan one time. It did it more than once. There was a first insect, a first bird, and a first mammals. So assuming that each first one was short-lived, it extended it, or vice versa. But anyway, it has been it's something that can evolve. So I think that it's pretty premature to say that just because by changing the exact same genes, a few of the same genes that we've changed in these worms in mice, and now there isn't 80. By the way, the worms that live 6 times as long as normal, 6 or 7, those worms have 2 changes, not just 1. And when you put 2 changes in mice that equally alone extend lifespan no more than 50%, now you get an 80% lifespan. But hardly any changes have ever been made in a mouse, and the only ones that have been made are made because of these works, pretty much because of the work on these little animals. So there's no science of this. This is just, we haven't, and to invoke evolution, I almost laughed, even though I respect you hugely, because that is exactly what people always said about the worm. All these evolutionary arguments, so that it was impossible to ever find one gene. And now we realize that in mammals, there's some new evolutionary argument that I actually don't know that prevents it from living very long. So I just think that well, we know that FOXO regulates all these genes, chaperones, DNA repair, antimicrobial genes, metabolic genes. We don't know what the potential of any one of those 100 downstream genes is on their own, if optimized, just by itself, is to extend lifespan. I mean, one of the things DAF2 mutants do is clean up the cleaning system that you just heard about. So if you do that plus many other things, you don't know. I'm not trying to say that we'll be able to live forever by doing what we've been doing, but I don't think you should discount it and say, well, it's all this reason for pessimism, because that's nuts. Okay, so that's the first thing. And what was the real question that I'm supposed to be answering? I can't even remember.
What's the timeline?
Oh, the timeline. Yes.
That's
That's
Parkinson's. You've been told.
It's about Alzheimer's. I may have it. Anyway, the timeline. Okay, so the cool thing. Here's what's really cool. Well, one of many real cool things. I just told you that these diseases, these interventions that delay aging also delay a lot of age-related diseases. Now, there are a lot of people out there already studying age-related diseases, trying to figure out what causes them and how they can counteract them. So you could imagine that by studying an age-related genes, you could bump into an aging pathway without knowing it. And you could make a compound for that disease, which actually delays the disease because it delays aging. Or at least it may also delay aging. And for example, people have IGF-1 is known to be the overexpression of IGF-1, the very hormone that I talked about, whose lowering extends lifespan. The overexpression of that causes cancer, or is a big contributor to cancer. So there are already many companies that are trying to make interventions to lower IGF-1. So they may, I don't know, I don't think that we're taking those pills yet, but the interventions are in clinical trials now. There are people that study centenarians. This is really cool. There are families of people who look like ordinary people like us, but they actually have changes in genes that they're kind of like a DAF2 mutants in a way. In other words, there are whole families in which it's very likely that people will live to be 100. And so in other words, these people, they're people, but they have different forms of genes that are passed on to their children that make it much more likely that they'll live to be 100. It's really interesting. So people are trying to map these genes and figure out what they are. And your Barzilai, for example, has found that one of these genes is called CETP. It's a gene that's involved in cholesterol transport. And Merck has already got drugs in clinical trials, late-stage clinical trials right now to target CETP for vascular disease. And so far, there aren't any terrible side effects that we know about. Now, we have no idea how effective this change would be on its own, because this may be one of many genes that's different in a centenarian family. But still, it could be approved in the next couple of years. So we don't know how effective any of these will be in people in terms of the postponement of aging. But we'll know something before too long, because people will be taking them. And then there are a lot of people, like my lab is now looking for small molecules that will activate FOXO, and lots of other labs are trying to do that approach with the things that they're working on. So, it could be that it will take a little longer. The big question is, how long would it take to make how big will the effect be? And of course, it seems likely that it will just be maybe 5 years, 10 years at the beginning. Although I can't rule out the possibility that we could get a doubling, because I saw it with my own eyes with DAF2 mutants, and no one thought that would be possible. So it's not out of the question, but it doesn't seem like the most likely scenario. And then it's also possible that for some reason people are just not going to be susceptible to any of these things. So that we don't know that they will be. We just don't know. Probably they will, but we don't know for sure.
Yeah, I really agree with everything that Cynthia's just said. In fact, first I want to just reassure Cynthia that I completely agree that we don't know whether it might be possible using simple interventions, changing one or two genes, using one or two different drugs, to actually get substantially more life extension and postponement of age-related ill health in mammals than we have achieved so far. I mean, at the moment, my intuition is that that's relatively unlikely, that the combinatorial approach, so to speak, does rather rapidly hit diminishing returns. Even in the nematode worm, actually the most extreme postponement of aging and increase of lifespan that's been achieved quite recently is a tenfold increase, is actually with making a change to only one gene. But absolutely, we need much more work in this area, especially in mammals. These experiments are hard to do and they're expensive, and that's why so few of them are being done. But there are people with purse strings who can change that. So I absolutely, completely agree that we definitely need more of this work. And I want to emphasize again that especially we need more work on late onset interventions, whether simple or perhaps slightly less simple. In terms of timeframes, I think, again, the sooner we get results in the laboratory, the sooner we'll get results in humans. When spectacular things are achieved in the laboratory, especially, as I said again, in late onset interventions started in middle age, people get pretty excited pretty quickly, and things move to clinical trials much more quickly than they otherwise would. As an example, let's talk about Alzheimer's disease for a second. Now, Alzheimer's disease is a very complicated disease with lots of things that go wrong, but one of the things that goes wrong, the accumulation of molecular garbage, not inside the cell, the sort of thing that Anna Maria was talking about, but actually outside the cell, in the spaces between our neurons. That sort of garbage, a lot of people who work in Alzheimer's disease think that it might be a good idea to get rid of it. And an approach to getting rid of it, not just to stopping it from being created, but actually getting rid of it, was demonstrated in mice in a paper published in 1999. That got to clinical trials in about 2 years flat. It was just ridiculous how quickly it went to clinical trials, and it's now in phase 3. So, we could move pretty fast if we really want to. Just a word about regenerative medicine, because Emily mentioned Parkinson's disease in the question. Parkinson's disease is mainly caused by the rapid death and, of course, non-replacement of neurons in a particular part of the brain called the substantia nigra, with the result that there is a deficiency in the production of a chemical called dopamine. Now, I would say conceptually obvious way to fix that in principle is to put back some neurons so as to reverse the problem, to repair the problem. That is straight up regenerative medicine for an aspect of aging. And indeed, there is clinical work going on to do exactly that, to put precursor neurons into that part of the brain so that they eventually develop into replacement neurons. And the best results that have been achieved in this way are by far the best results that have been achieved in any way in treating Parkinson's disease. But those results are not achieved consistently at this point. Some people have tried this and it hasn't worked. Some people have tried it and it has worked. And it is, to my mind, utterly appalling that the inconsistency of these results is resulting in lack of enthusiasm for further pursuit of such work. Because it's normal in science. As Einstein said, if we knew what we were doing, it wouldn't be called research. It's normal in science for things to be inconsistent early on, but this is by far the most promising approach to treating people who've already got Parkinson's disease, and it's an example of the application of regenerative medicine to a very important aspect of aging. It needs a lot more support.
I'm going to open it up now for questions from the audience, and we have, Sean, do we have someone with the Sean Hayes of New America Foundation is going to hand the mic around. Your hand went up first, back. Yes, right.
I'll come forward. I'm Jason Robert. I'll get to talk later, so I'm just going to make this really quick. I want to follow up on the point that Aubrey was just making about regenerative medicine and Parkinson's disease. There's some really interesting data that have been gathered in this, especially working initially with fetal ventral mesencephalic cells, which are a particular kind of cells that are harvested from aborted fetuses. Not a sort of particularly easy way to get the cells, and the work has been pretty uneven. Some of the results are absolutely spectacular. I used to work with one of the neurosurgeons who did this work, and he was really successful at it, but very few other people were. So the thought was, let's move this on to deriving precursor cells from other sources rather than from the fetuses and see if that works. And there have been a lot of obstacles along the way to getting that done, as Aubrey just mentioned. But one of the ones that still resonates for some people is that maybe it's the case that replacing dopaminergic neurons is only part of the issue, and that regenerative medicine is focusing just on that one piece, and we need a more systemic approach to figure out what's going on with that disease more broadly so that we can either maximize the benefits of regenerative medicine or turn our attention elsewhere.
Yeah, I
Wait a minute. I can tell you're a philosopher because that was a short question.
Yeah. So I completely agree. And the thing I want to emphasize is that the only really effective way to determine whether a particular treatment for a particular aspect of aging, or indeed for any complex medical problem, is a comprehensive treatment, is to do it and see what happens, see what's left. If it doesn't work, or if it only partly works, then we have effectively unmasked the remainder of the problem that we could not actually explore and investigate properly when the part that we did understand was still in place.
More questions?
Thank you. I'm Dan Perry. I head the Alliance for Aging Research here in Washington, and I've been at this for quite some time. And I think most of the panelists will agree with me that in recent years it's really become dogma among the biologists that do research in aging that aging is malleable and that it can be changed, and to our great benefit because it will keep people healthier longer. And this is what we've heard from a lot of the panelists. But Aubrey said something very important when he said that it will be when the benefits are anticipated in the political realm that there will be a payoff for the political support for this. And if this is now dogma among gerontologists and biologists and aging and in many, many meetings like this, it is not talked about when you go to the American College of Cardiology, when you go to the American Cancer Society meetings, when you go to the Society for Neuroscience. And those are the meetings that are intensely covered by the media. And so that's where the anticipation of applying modification of aging to reduce disease will take off. And my question to the panelists is, when will that happen? And what will it take to bridge this new orthodoxy from gerontology and research in aging to the larger medical world?
So let me answer that a little bit. So some of the work that we are doing on early detection of disease was actually funded by DARPA. And
explain what that is.
DARPA is the Defense Advanced Project Research Agency. They're the guys, the stealth bombers, the ARPANET, all that kind of stuff. And so they funded this. I forgot what I was going to say. They funded this in terms of biological warfare detection. And so and when we were telling them about the stuff we were trying to develop, we realized that we would never sustain something to protect us against biological warfare unless we connected it to something was done as standard clinical practice anyway. So anything we did had to have a connection to standard clinical practice. Otherwise, it would be like BioWatch. It's an unsustainable system. And I believe the same thing about the aging. The aging initially will probably come out of not saying, let's have this big project and spend more on aging research and things like this. It'll become because we connect it to the healthcare crisis. So any research that we have, as much as we can connect it to solving fundamental problems in the healthcare, that will be funded. And that's why we've been going after pre-symptomatic diagnosis. And it may well be that by doing that, you also will substantially change life extension or the quality of life because you're detecting disease early. Not in a way we may know how that will be now, but it's unpredictable. And same thing with a cancer vaccine. We think cancer vaccine may have effects well beyond cancer because you're taking out inflammatory cells. And so if you're taking them out fundamentally, you may extend the life expectancy. So those mice that we're trying to prevent cancer in, we're letting live to see how long they live. Do they live longer? So, and back to this other issue on the time frame, and this is a little controversial, maybe more than a little, but I think we're not going to see, we won't know those time frames unless, what we could do with it, unless we fundamentally change how we do biomedical research in this country. We've done this, our portfolio now is basically investor-initiated research. It's not mission-oriented. It's not Apollo project type projects for whether it's on aging or cancer or anything. We can announce we're a war on cancer, but we still just distributed the money to everybody and said, we hope a solution happens. We're at a point now that we should start rethinking that whole process. If we do, we've done a lot of that seeding work already. As a country, we have to start rethinking how our portfolio for an investment in this area. Otherwise, it's going to take us a long time to see any of these things happen. But if we start getting a little focused here and change our portfolio, our investment portfolio, I think things could happen quite quickly.
Speaking of time frame, I'm going to...
Can I disagree with that, though? I fundamentally disagree. That is really... it's sort of like saying the guy who said there will be no more discoveries, right, in the end of the 1800s. The patent office head said, that's it. We've invented everything. That's it. And that's like what you're saying. I think that we should... Wait, I think we should have... I think it's good to have... We already have foci. We already have a cancer project. We already have projects. But
of the NIH funding goes
would you please be quiet so that I can speak? I was quiet while you were speaking. Thank you very much. I would never have done my experience. I worked on developmental biology. I wasn't working on aging, and I just had this idea that that they were wrong, and that there would be genes for aging. So I looked. It was just an idea, and I was successful. It didn't come from, a project from the government. And I think that it's good we do need to focus somewhat. But if we don't have curiosity-driven research, just people in the lab just doing whatever they want to do, that's how lasers came along, that's how lots of things came along. If we cut that off, I think that it's very dangerous. I do. Especially, the more, if you have to justify every single experiment you do as being right, it's like turning NIH into a pharmaceutical company, and I think that's very dangerous. So I just want to have a little balance.
So this is the typical NIH response that you get from an NIH researcher.
Stefan, speaking of time frames, I'm going to cut this off because we've got so many questions, and we've got limited time. Young man.
Aren't there multiple variations of resveratrol? I think the one that's most appropriate for human absorption is trans-resveratrol. The typical supplement, I think, is a 50-50 hybrid of trans-resveratrol and standard.
As I recall, yes. There have been several investigations looking at the differences between using racemic mixtures of 50-50, as you say, as opposed to the two different stereoisomers, and they do have differential activities. I don't know what's available commercially at the moment in terms of things that are only one of the different stereoisomers. However, I do know that the somewhat limited effect, limited to mice with a high-fat diet that Cynthia mentioned earlier, it's work that has been done with with all of these variations, and it doesn't seem that just by using only one stereoisomer we can get dramatically better results.
Was there? Yeah, over here. Yes.
Thank you. Thank you. Sterling Wright, I'm the Director of Global Future Media. Ultimately, my question will be addressed to Mr. de Grey, and I'll be very brief. I was very happy that the gentleman, Mr. Call, who opened this presentation, gave credit to the Department of Defense for the work it's doing in this research. And if there's anything that serves as a greater impetus, if there's any upside to being actively engaged in warfare, it's the impetus to cure the maimed soldiers who are returning from the battlefield, so we can thank the DoD on this front in many cases. Mr. de Grey, you've expressed a great deal of pessimism that members of On the Hill would be supportive of research into regenerative medicine, and yet at least that's what I understood. Okay, is that correct?
Not exactly, but go on.
Not quite, but okay. And we certainly have heard in the popular media about tremendous advances that DoD has made. For example, extracting and distilling compounds to regenerate damaged muscle and growing entire structures like heart valves, etc. And those representatives on the Hill who are warrior-friendly, so to speak, are very supportive of this. So again, I'm curious where your pessimism comes from.
Sure.
Thank you.
So yes, the only thing that's not quite accurate about how you heard what I said is I certainly don't think that regenerative medicine in general is a hard sell. I think regenerative medicine applied to aging is a hard sell simply because aging is this extraordinarily complex, multifaceted degenerative phenomenon which will only be treatable by regenerative medicine if you have a lot of different aspects of regenerative medicine all developed and applied at the same time to the same people. And that's why it's going to take a long time to develop. It's the time frame that makes it, less relevant to the political elite. In the case of regenerative medicine for wounded soldiers, for example, you're absolutely right. This is something which has a great deal of priority and which is something that can be developed in, we hope anyway, in a relatively short time frame. I want to mention DARPA again, which Stephen mentioned. DARPA is an extraordinarily important player in all of this. They, as the A for advanced indicates, they are interested in relatively longer term, more blue sky research. And it's very, very important work, and certainly a lot of the people really at the cutting edge of regenerative medicine are funded by DARPA. In fact, that's a fine way to allude to the misunderstanding that just took place between the people on my right, because I think that there is no debate at all about the value of curiosity-driven research. However much of an engineer or a technologist one may be, there is no disputing whatsoever the absolute necessity for curiosity-driven research, and I'm quite sure that Stephen doesn't think any differently. But we definitely also need a balance. We need to have the right amount of work in translational research and development, and sometimes it's hard to tell exactly when the balance should change. Because, of course, there is a point when the basic knowledge that arises from curiosity-driven research reaches a level that can be exploited in a goal-directed technological way, and sometimes it's very unclear when that point has been reached, and it's not always the people who are best at the curiosity-driven research who can necessarily tell. It takes a different sort of thinking to do technological work than it does to do basic science. So we need to be constantly revisiting that question. And certainly the concept of applying regenerative medicine to aging is something that has been much more readily embraced by people who work in regenerative medicine than it has by people who work in aging.
Aubrey, I'm going to cut you off because we have only 10 minutes. Yes, sir. And please identify yourself and make your question succinct because we're running out of time.
My name is Richard Benedict, formerly State Department President of the National Council for Science and the Environment. Quick question. Mention was made, of course, of physical and chemical interventions that are still underway in the laboratories. My question is that also mention was made of glucose and fat. While these scientific interventions are being researched, what importance would you assign to, in the near term, directed nutrition as a factor in this whole scheme?
What is directed nutrition?
Conscious nutrition. I mean, not just following the advice of nutritionists and scientists, saying what's good for you and what's not, and would that be important or not so important?
I actually think that Cynthia really
I can answer that in one question. It is unbelievable that we don't know what to eat. Isn't it amazing? We don't know. I mean, when I see a hamburger, I want to eat the hamburger without the bun, right? But a lot of people want to eat the hamburger without the meat or eat only the bun. I mean, it's nuts. We don't know what to eat. And I think that's shameful. And I think that's behind this, at least partly behind this horrible epidemic that we have in obesity, even among children, and diabetes. It's horrible. And I think that's very, 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 very important. I also think that's going to be a very highly respected and exciting field to work in the next few decades, unlike the way it's been sort of sidelined in the past few decades.
Over here.
I'm Stephen Schafferman, And I'm curious that there's been no mention of fitness, exercise, and movement. I work in a new approach to fitness and movement based on the way young children learn and move, and we're getting marvelous results in reversing arthritis, hip degeneration, even restoring the harmony of the nervous system for neurological conditions. So I'd love to hear your comments.
There have been a lot of studies done on the value of exercise among the elderly, and it's amazing. You can take people who are in the nursing home or almost in the nursing home and a little resistance. So now, they're not very strong. They can't lift very heavy weights, but they can lift something. And if you give them just a little less than the maximum amount that they're able to lift, so suppose the most they could lift would be 3 pounds, you give them 2-pound weights, and you let them lift them and walk a little bit more. The change that you see in these people, even the cognitive changes, are amazing. I'm not saying that they're going to live, you're not making them younger, but the change is really, really striking. I think there have been studies asking whether exercise per se extends lifespan, and I don't know of any case in which it did, but the health benefits and the cognitive benefits are very large, and I exercise, and I definitely think it's a great thing to do.
Anne Marie.
Unfortunately, I don't exercise, but I still think it's very important for the people who wants to live longer. And just to say, there is a lot of molecular basis now for the exercise. We can give numerous examples, but even things like these chaperones, for example, that we were saying identifying damage. There is a very nice study done in humans showing that elders who exercise have a better response of these chaperones, so they will be able to sense damage better. And that's just a tiny example of the many beneficial effects. You are balancing your energy, you will modulate the consumption of the fat stores that you have, so that's going to have a major impact. And I think along with nutrition, exercise is also one of the areas that has to be emphasized.
Just very briefly, I completely agree. And I think the only reason you haven't heard about it today so far is because it's really not the focus of this conference. The focus here is on the development of things that might solve problems, of technological advances. Whereas, we have found that it's pretty difficult to get people to exercise when they should, just as it's difficult to get people to eat less when they should.
Back there. Lestro.
I need you to come forward, I'm sorry, in order for the microphone to work.
Yes, my name is Rob Nissen. I'm just a complete layman. My opinion was that the longevity research kind of came out of the observation that calorie restriction appeared to greatly lengthen lifespans in worms and then on up the food chain. Is the research going away from that? I thought the original idea was to see what can we do chemically or by a genome to mimic the evident the increase in life expectancy of calorie restriction. Is that still a prominent area of research, or is that going away?
Well, I started our experiments without thinking at all about caloric restriction, just thinking everybody's ignoring aging as something that would be controlled by the genes, and everything else is controlled by the genes, why not aging? And we found these pathways that affect aging, and they turn out to be pathways that affect nutrition and could converge with the caloric restriction phenomenon. But I think they're related, but I don't think they're necessarily totally congruent. And I think as we know more, we'll be able to appreciate that more. I think that I certainly don't think that all we're doing is trying to find ways of mimicking the response to caloric restriction without going hungry, even though some of the pathways that we find may be doing that.
Okay. Yes, over here.
Yeah, at the risk of Mario Arancibia, Chief Technology Officer of Merlin Telecom, but basically a layman worried about aging for personal reasons. At the danger of sounding a little bit reductionist and superficial, I have a few notes here. It's going to be short. I think we should take a hint from the visual indicators of aging in terms of how to proceed in a fundable manner. Understanding and Aubrey de Grey indicated that aging is an extraordinarily complex and degenerative process. And I believe that baby steps here will help. I propose founding the Cure Gray Hair Foundation. It is For several reasons. It's a very atomic way of facing aging. Everybody would understand what the goal is, to reverse gray hair. It can probably get funded very easily, given the established market demand for
Not
color,
by the
hair
hairdressing
coloring.
industry, however.
Right. Well, if you convince them that they can make more money, the results may spill over to understanding and reversing other type of cellular damage. And it is RM. It is regenerative medicine.
I'm going to take that one. That would save me so much money, and I hope your project gets off the ground. And let's take one last question. Yes, orange in the orange shirt.
My name is Naoto Oka. If we can control the process of aging better than what we do today, let's say we live on average 15 years or longer, what happens at the end of 15 years? Would there be other diseases that we don't know yet today, or can they die happily?
Yeah, let me. I think that's a really good question. If we don't extend life 70, 100 years, but we get 5, 6, 7 good more years out, what is going to get us in the end?
That will be interesting to do. And I think we have to start thinking about this whole thing sort of as our big evolutionary experiment. I'm not sure what will happen. If we start detecting diseases early and we don't have cancer, we don't have diabetes, we don't have these other things, what happens to our life expectancy? And as alluded to here, do we start having a square or life curve where everybody dies of a stroke at 125? Or is it just that we've pushed things out and then the length of decline is just longer, it's just pushed out a little more? I'm not so sure what will happen, but I think it's going to be a fascinating experiment to do.
We don't know really. Well, there are precedents for both. It's from animals. So there are 2 long-lived mouse mutants, one where the insulin receptor is removed in the fat tissue, and another where one of the downstream proteins in the insulin pathway called IRS2 is mutant. And in both of those mice, they live long, they look great, they look young, and then they die. But they all die very suddenly, and no one really knows what they die of. It's kind of perfect, you know? I mean, really, isn't it? But then there are others, like certain changes that you can make in the worm that keep the worms young, and it's really like aging, taking 2 days to age as much as you'd normally age in one throughout the life. So they stay young and healthy longer, they have a longer middle age, and then they do have a longer period. So now maybe if the person's in the nursing home, they wouldn't be allowed to take the pill anymore. I don't know. But we don't really know yet why these are different. These are subtle things. I mean, they're important, but we don't understand them molecularly yet. But there at least is precedent for both, which is good. It's good.
And that seems like a perfect place to stop. Thank you all so much, this marvelous panel. We're going to take a coffee break.